all, Miss Psilo's here to teach you about mitosis, cell division in our body cells. This may be a review from middle school, when you should have learned this in the past. We may introduce a few more details this time, though. For this lesson, we'll answer the following questions. 1. What is mitosis, and why and when do our body cells use it? Number 2. How does the DNA change during cell division? And 3. What are the steps of mitosis? During the lecture, make sure you take detailed handwritten notes. A note-taking guide should have been provided to you by your teacher, and it's optional to use in order to organize your notes. But you don't have to use the guide and may still take notes in whichever manner or style that you prefer. Let's begin with the basics to answer our first question. What is mitosis, and why/when do our body cells use it? Mitosis is cell division that takes place in somatic cells, and it results in two genetically identical daughter cells. Okay, I just used a lot of new terms in our definition here. I will get to the somatic cells on the next slide, but what does it mean by genetically identical cells? The two daughter cells created at the end of this process should have the exact same DNA, the same amount of DNA, and the same types and amounts of organelles. They should be clones of each other. So genetically identical means that they have the exact same DNA, and they're the same ultimately in every which way. The other term, daughter cells, just refers to the two cells that are the products of cell division. The original cell that starts out the process of mitosis is called the parent cell. After that parent cell goes through mitosis and splits into two, the two cells that result will be called daughter cells. How about somatic cells? What are those? Well, if we look at a multicellular organism, it is composed of hundreds of different types of cells. We can categorize the types of cells into two major categories. There are somatic cells, which encompass our body cells, like skin cells, nerve cells, blood cells, muscle cells, intestine cells, bone cells, skin cells, etc. I think I said that one twice. Yep, I definitely did. But anyways, you get the idea. Somatic cells are the cells that allow the organism to function normally in its daily life. The second category is called gamete cells. The only types of gamete cells in multicellular organisms are egg and sperm cells. Gamete cells are those that are used for reproduction, to create a new multicellular organism. So, our definition of mitosis one more time. Mitosis is cell division that takes place in somatic cells only, resulting in two genetically identical daughter cells. Gamete cells use a different type of cell division, and we'll come back to what that is exactly in another week. Okay, so why is it necessary for somatic cells to do mitosis? Or why is it necessary for them to divide? What's the purpose? Well, cell division in somatic cells is important for two things. It helps the organism grow, and it helps the organism repair its tissues. When a multicellular organism needs to get larger, the only way it can do so is by adding more cells to its structures. It's like a brick wall. If you need that wall to get bigger, you simply add more bricks. If an organism needs to get larger, you need to add more cells to extend the length and width of the tissues that compose their body. Cell division is also needed to repair damaged or dead tissues. Let's say your brick wall has a crack, and the integrity of that wall has now been reduced. It shouldn't stay like that. To repair that wall, you could remove the damaged area and replace the hole with new bricks. Same idea. If a tissue in the body is damaged, the area of damage could be removed, which would result in a hole or a gap in that tissue. And that hole or gap would eventually need to be filled in. New cells would refill that hole, allowing the tissue to heal. Take a look at our gifts here. When we grow, our structures do physically get larger. They may change in shape too as we go through development. New cells will need to be made to allow for this. Same deal with our cut here that you see healing in the GIF on the bottom. Over time, new cells are replacing the dead or damaged cells that are within that cut area. As time goes on and new cells are made, that area of skin will look good as new. Because it is. Next, when do cells carry out mitosis? Well, we need to take ourselves back to the cell cycle. Remember that? The cell cycle is the process cells go through when they prepare and then carry out cell division. In the cell cycle, there are two major parts. We have interphase and mitosis. Interphase is the part where the cell will prepare materials, copy its DNA, and grow a bit in size to prepare for division. G1, S, and G2 phases. Mitosis is the part of the cell cycle where the cell actually divides. M phase. So we're here in the cell cycle that you see in this blue highlighted area as we talk about the process of mitosis. If you need to pause the video to review your notes on the cell cycle, you should do so before moving on with this video. Okay, so let's move on to our second question for this lesson. How does the DNA change during mitosis? Now, before we get into this, let me preface by saying that I don't mean the DNA code is going to change. No mutations will purposefully take place during this time. By change here, I mean that the DNA's shape is going to be a little different during mitosis. So typically in a cell, we can describe the DNA as a long string. It's an extremely long molecule. And in humans, there are 46 DNA molecules in every cell's nucleus, which means we can imagine 46 very long strings stuck within the small, finite space of the nucleus. How do they all fit without getting tangled and knotted around each other? I'll answer this question in a sec, but let's focus on what happens during mitosis. At the very beginning of mitosis, the DNA's shape is going to change. Looking at the picture here, we can see that before mitosis, the DNA is very long, loose, and stringy. During mitosis, the DNA condenses into a very tight and smaller shape. Why might it need to be condensed into this tighter and smaller shape? Take a look at the diagram, which shows the steps of mitosis. On the left, we see a cell in interphase, a cell that has not started mitosis yet. In the second picture, we see that the cell then begins mitosis, and its DNA has changed shape to that smaller version. What happens to the DNA during mitosis? Do you notice that the DNA is going to be moving as the mitosis steps progress? In order for the DNA to not tangle and break as it's being moved around the cell, it's helpful to condense the DNA into a smaller and tighter shape. So the DNA needs to be condensed at the start of mitosis because it will be moved around the cell during the process. It will line up at the center of the cell and then move to opposite ends of the cell as the cell is dividing. So condensing the DNA reduces the chance of that DNA breaking or getting damaged during this time. In order to describe the different shapes that DNA could be in, we use specific terms or names. The long, stringy form of DNA that we see here on the left, that's called chromatin. In your notes, you should underline the word chromatin in the same spot that I have underlined it on the screen. This will be helpful, and you'll see why in a bit. Chromatin DNA is seen at all times except for when the cell is going through mitosis. The very condensed, tightly wound, smaller, and even thicker form of DNA that we see here on the right, that is the DNA shape that we only see during mitosis, and it's called a chromosome. So we only see DNA in chromosome shape during cell division. On the left now, in this picture on the left, we have a cell that is still in interphase, meaning that it has not started mitosis yet. We can see that its DNA is in chromatin form. However, at the very start of mitosis, the DNA will be condensed into chromosomes. If we look under the microscope during this time, when the cells are going through mitosis, you can clearly see chromosomes. The DNA in cells have condensed so much that they're easily visible under the microscope, and I'm pointing to an example of this on the left here. So those are chromosomes, very thick, dark lines. That's what it looks like under the microscope. The cells that are not actively going through mitosis, though, they have chromatin DNA. So their DNA is not so easy to see inside of the nucleus. And I'm showing an example of that on the right, what I'm pointing to on the right here. So how does the DNA get condensed anyways? What makes it become smaller and more tightly packed? Well, in general, DNA is wrapped around these proteins called histones. In our GIF here on the left, a histone is represented by this purple blob. The blue string is the DNA, and we can see that it wraps around these histone proteins just like if you wrapped a piece of string around your finger. When the DNA wraps around histones, it gets a little bit smaller or condensed. You can see that happening in the GIF too. So DNA is always wrapped around histones. That's why 46 long strings of DNA can fit normally inside of the nucleus. How is this coiling different during mitosis, though? Well, during mitosis, the histone proteins will wrap the DNA even tighter. This makes it condense even further, changing the chromatin DNA into chromosome DNA. Now, there are two ways that we might see chromosomes, or two types of chromosomes that we might see during mitosis. Each of these are considered to be just one chromosome. So this is one chromosome, and this together is still considered one chromosome. Get it? Okay, so what's their difference? If you see a chromosome looking like this, then you're looking at an unduplicated chromosome. It's a single DNA molecule that's been supercoiled into the chromosome shape. We can simplify the DNA image to look like what you see on the right. It's a single DNA molecule that has not yet gone through DNA replication. If you see a chromosome looking like this, though, you're looking at a duplicated chromosome. To make a duplicated chromosome, a single DNA molecule had to go through DNA replication so that a copy of that DNA molecule could be made. So in a duplicated chromosome, we are seeing two copies of DNA connected together. So here's one copy right there, and here's the second copy. And these two copies should be exactly the same to each other. Even though in this duplicated chromosome, there are two DNA molecules, when they're connected together like this, the whole is considered to be just one chromosome. So let's compare the two side by side. On the left, we have an unduplicated chromosome. If a single DNA molecule is condensed into its chromosome shape, it'll look like this one on the left. Okay? Now on the right, we have a duplicated chromosome. Imagine that a single DNA molecule that we had was then copied through DNA replication. In that case, we'd have two DNA molecules, each one being a copy of the other. And that's what we're looking at here with the duplicated chromosome. There are two copies of DNA connected together at the center, this part right here where it's pinching inwards. One copy, two copies. When was the DNA replicated exactly? At what time did this take place? Well, if we don't remember, DNA replication takes place during the cell cycle in S phase of interphase. S phase takes place before mitosis begins. So that means all the DNA in a cell will be copied before mitosis gets started. This is a preparation step that must take place before mitosis because each daughter cell created at the end of mitosis will need to receive its own copy of the DNA. Hopefully, when DNA replication occurs during S phase, that no mutations are created in either of the copies. If mutations do happen, the cell cycle checkpoints should ensure that those mistakes are fixed before mitosis begins. So like I said, the reason that the DNA needs to be copied before mitosis begins is because each daughter cell created by cell division needs its own copy of the DNA. That DNA that each cell receives should be exactly the same or identical. No mutations, no mistakes in either one. If so, If there's no mistakes, then each daughter cell that receives that DNA should be genetically identical. Sorry, genetically identical or a clone of each other. One more thing before we move on. Let's take a look back at our duplicated chromosome. Like I said, a duplicated chromosome is composed of two copies of the same DNA attached together. We can label these two copies of DNA in a duplicated chromosome. Each would be called a chromatid. In your notes, you should underline the word chromatid in the same spot that I have underlined it on the screen. So this is one chromatid right there, and this is another one. Or if we're looking at this drawing, this is a chromatid, and this is the other one. So they're only called chromatids when they are attached together in a duplicated chromosome. When the two chromatids are connected together in a duplicated chromosome, we call them sister chromatids. Alright, so with that information, we're now ready to go through the steps of mitosis, which will answer our third question for the lesson. On the screen, you should see a cell that has gone through interphase, meaning that it has grown in size, doubled the number of organelles, replicated its DNA, and created this organelle, the special one called centrioles, that will be used during mitosis. If all appropriate materials are created and the checkpoints have been passed, the cell is now ready to start mitosis. Here are the four major steps that will take place during mitosis. To help you remember the order of these steps, I want you to memorize PMAT. P-M-A-T. Make a little note somewhere on your paper that this acronym will help you remember the steps in order. P-M-A-T. PMAT. So those are the steps of mitosis in order. The first step of mitosis is called prophase. P for prophase. Take a look at our diagram. What do you see happening? First, we can see that the nucleus that surrounds the DNA is being dissolved. This will free the DNA from its cage, allowing it to begin to move around the cell. We can also see that the DNA has been condensed into chromosomes. And we can see duplicated chromosomes here, right? Because S phase occurred before mitosis got started, so all the DNA should have already been copied. That's why we see duplicated chromosomes here. Lastly, we can see that the centrioles, that special organelle that was created during G2 of interphase, we can see that they have migrated to opposite sides of the cell and are starting to create these little string-looking things. These are called spindle fibers, and they'll have a job to do during mitosis. The second step of mitosis is metaphase. M for metaphase. Again, look at the diagram. What happened from prophase to metaphase? What changed? Here you should see that the duplicated chromosomes have moved to the center of the cell and are lining up one by one on an imaginary center line of the cell. A helpful hint to remember what is happening during metaphase is this. M for metaphase, M for middle. The chromosomes line up at the middle of the cell during metaphase. Maybe write that little tool down to help yourself. We will also see during metaphase that the spindle fibers have gotten a lot longer and are now reaching out to connect onto the duplicated chromosomes. These are like little lassos that latch onto the chromosomes. Before progressing to the next step, the cell will pause to conduct a checkpoint. It's the M checkpoint that we learned about before. During the M checkpoint, the cell will, quote-unquote, ask itself the following questions. Number 1, are the chromosomes lined up appropriately in the middle of the cell? And number 2, are all the chromosomes attached correctly to the spindle fibers? If the answer to either of these questions is no, then the cell should not continue on with mitosis until the issues are fixed. If they're never fixed and the cell continues, you might end up with two cells that are different from each other. One of the two cells might have more DNA, while the other cell has less DNA. Or one cell might have more organelles than the other, making it a little bit larger than the other. The spindle fibers should be separating things equally so that the two cells made at the end of mitosis are exactly the same in every way. If the checkpoint finds no mistakes, no errors, then mitosis should continue. The third step of mitosis is anaphase, A for anaphase. During anaphase, the spindle fibers will now start to pull backwards towards the centrioles, towards the opposite ends of the cell. This will split the duplicated chromosome apart, or will pull the sister chromatids away from each other. Remember, in a duplicated chromosome, you have two copies of DNA that are attached together. So if the duplicated chromosomes are being pulled apart, that means each copy of the DNA is being pulled away from each other. As another hint to help you remember what happens during anaphase, think of the following. A for anaphase, A for apart. The duplicated chromosomes are being pulled apart during anaphase. The last official step of mitosis is telophase or telophase. We can see that the cell is beginning to pinch at the center, which is starting to form our two daughter cells. However, the cells have not yet completely pinched off from each other. They're still attached in this area in the middle. We can also see that in each portion of the cell, a new nucleus is starting to form, surrounding the unduplicated chromosomes. This left nucleus is holding one copy of the DNA, and the right nucleus is holding the second copy of the DNA. If DNA replication occurred without any mistakes, these two sets of DNA copies should be exactly identical to each other. Lastly, we don't see any spindle fibers anymore. They have been broken down because they're no longer needed. Everything has appropriately moved. One more hint to help you remember what happens during telophase. Think of the following. T for telophase, T for 2. We are beginning to see two cells forming during telophase. What then splits the cells if telophase doesn't? Well, telophase is finished by an action called, when telophase is finished, an action called cytokinesis will occur. It's a really quick action, so it's not considered a step of mitosis. During cytokinesis, the cells will completely pinch off from each other, separating into two independent cells. These are your two daughter cells that result from mitosis. What else do you notice about the DNA? You should not see chromosomes anymore. During cytokinesis, or maybe even just before cytokinesis, the chromosomes will loosen back up into chromatin. This is why we see long strings of DNA that are hard to tell apart. Okay, so by the end of mitosis, we have the following. Two genetically identical daughter cells. That means these two cells have the same organelles, and they each have their own nuclei with their own copy of the DNA. Their DNA copies should be identical, hopefully without mutations. They should also have the same quantity of DNA. So if these are human body cells, let's say skin cells, they should each have 46 DNA molecules in them. If it's any more or less than 46, then a problem probably occurred during mitosis. Alrighty, we're all done.